Dr. Kim got his PhD at Ohio State University and then did his postdoctoral training at Harvard Medical School. He's now the Dean's Professor of Microbiology and Immunology and the Director of the Tumor Immunotherapy Research Program at the Wilmot Cancer Institute, and also a member of the Smith Center for Vaccine Biology and Immunology. And his lab over the years has really been pioneering imaging techniques to visualize immune responses both in live cells as well as in vivo. And he's thinking about a paper called efferocytosis, T-cell immunity to influenza infection, which was published last year in August. So Dr. Kim, over to you.
All right, thank you very much. Let me try to share my screen. All right, so sorry. Oh, anyways, okay. Thank you very much, everyone. I think this is a great idea. Having this meeting like this to share your research progress with so many people around the world, especially during this difficult time. So thank you for the doing this and also for the invitations. Ivan presented beautiful study with neutrophils, and actually he covered a lot of sort of basic concept already. So, but the paper we published last year was actually the extension of our original paper published in 2015. So I thought we could actually combine these two stories together so that I can help you to understand the overall study. So we study influenza infections, especially using a mouse model. I mean, just like any type of respiratory virus, just also like COVID, the virus are mainly coming into the airway and start to infect upper side of the airway, including trachea, and this is highly pathogenic. Then it will start to spread to the lung. In our lab, we use a technology called a multiphoton microscope or two-photon microscope. By the way, do you see my cursor here? Is that okay? So this is our mouse experimental setup. So we can actually intubate mouse through the trachea so that we can keep a mouse live, and at the same time we can immobilize this live trachea so that we can image host immune response, sometimes 6 hours or 7 hours. With this mouse model, one of our first question we had was, we have so many different type of immune cells, like innate immune cells and adaptive immune cells. The question was, how these different type of immune cells work in different time point. What's their kinetics, especially during this active infection. So we start to look at the different type of immune cells, starting from neutrophils. And neutrophils coming into the influenza infected trachea within hours, and they have peak like day 6. By the way, this mouse model is 100% recovery model, so the neutrophils near completely disappear by day 9, and the monocyte follow very similar kinetics with neutrophils. Unlike neutrophils and monocyte, macrophage initially drop a little bit and then very quickly repopulate, and also the same case for DC. So the one important information here is, during this massive innate immune response, virus titer doesn't really change a lot. Only when we have a very strong and specific antigen-specific adaptive immune response, CD8 T-cell response coming in day 9, and also the virus-specific CD8 T-cell response really happening at day 9. Because of that, the virus again and cleared, the mouse recovered. So that's our model. So once we set up these models, there's different type of immune response. One of the big questions we had at that point was, we have such massive neutrophil response, which doesn't directly clear virus. Why we need such dramatic neutrophil response, and what's their impact or contributions to this host immune response? So to answer the question, the most easy way or straightforward way is just erase neutrophil from this graph or delete neutrophil from the system. And like Ivan show in his paper, we use for our case we use an antibody called 188 antibody, and which can actually deplete 90% of neutrophil, up to 90% of neutrophils in circulation within hours, and then infect mice and look at the CD8 T cell response on day 7. So whenever we deplete neutrophil, the overall CD8 T cell response start to decline, and then especially this virus-specific CD8 T cell response was very significantly suppressed. And because of that, the virus clearance also very delayed. But it was not only the number we got. So this is mouse influenza infected trachea sections here. So in normal case, a lot of CD8 T cells come, but they really come directly to these infected epithelium, and they kind of obviously doing their effector functions. But if you deplete neutrophils and then infect mice, still small number of CD8 T cells can come, but their location was just near completely disorganized. They just couldn't get to their target location. So we decided to look at this distribution pattern a little more carefully, again in live influenza infected trachea. And this is top view and this is side view. And the outer area of the trachea is shown in blue color with second harmonic generations, and then infected epithelium is labeled in red colors. So in normal case, day 7, a lot of CD8 T cells come, and as you can see, many T cells make direct contact with infected epithelium. But if you deplete neutrophils and then infect mice, still small number of T cells can come, but none of these T cells actually can make contact with their effector target cell type. So the summary is just simple depletion of neutrophil from this picture not only change CD8 T cell response, but also the locations and their migrations, it was dramatically changed. So what's the contribution of neutrophils? So how neutrophil impacts such dramatically the T cell response? So we decided to look at neutrophil migration more carefully, starting with in vitro system. So this is neutrophil migration on several different substrates like ICAM1, collagens, or fibronectin. So if you look at their migration, almost all different type of substrate we tested, they actually leave very significant particles and very little trails behind in multiple times. But you can say, some extreme thing can happen in vitro setup. So we move back to the in vivo imaging. And again, even in before this neutrophil exit in inflamed tissue, they leave these multiple particles when they cross. And also when they exit blood vessels and coming into the tissue, many neutrophil leave small particles and trails behind. You can zoom in. So when neutrophils migrating following this collagen fiber, as you can see, they leave this multiple trails behind. So this really suggests that not only in vitro or in vivo, they leave these trails a lot. So we thought that maybe these trails might have some sort of signal, so potentially can guide later CD8 T cell response. So obviously the main signals can be delivered is chemokine signals, and neutrophil is actually known to express several chemokine in their body. But among those chemokine that neutrophil produced, the only chemokine, which in this case again CXCL12, was very highly enriched in neutrophil trails comparing to their cell body. And if you actually label CXCL12, a lot of chemokine is actually very nicely packaged in the particles that mainly localize at the tail of migrating neutrophil. So that could really suggest that maybe neutrophil trails contain important chemokine that could potentially guide CD8 T cell migrations or locations. So to prove that questions, to directly answer that question is we wanted to visualize T cells and neutrophils simultaneously in an inflamed tissue. So you see the red T cells and green neutrophils in inflamed ear tissue. And even neutrophil already left multiple particles behind. But if you look at the specific T cells in this case here, it touched first neutrophil particles here. and then it comes to directly the second neutrophil particles here, and then this T cell migrate directly to the third neutrophil particles here. So both, in vitro and in vivo, the data really suggests that just neutrophil trails can function as breadcrumbs, but in this case, neutrophil trails contain CXCL12, which is actually potentially very important chemokine to guide CD8 T cell migration for their location. So that really suggests that all the neutrophil response is actually quite important for the T cell migration and location. Once we published this data, one of the striking things that we couldn't answer from this study was the later part of this neutrophil. So even before we start to clear virus, or even before we have productive CD8 T cell response, that this massive neutrophil response was very quickly resolved and disappear. So what's the biological implication? Why we have such massive neutrophil, but very quickly disappear from tissues? What's the biological implication of this response? Well, that was our second question. There's so many different ways you can actually resolve or stop neutrophils coming into the tissue. You can shut down your production in bone marrow. You can shut down their entry to the tissue, or sometimes neutrophils can drain into other area, but none of these were the case. There's several recent suggestions that some of the neutrophils actually come and then to the tissue and reverse migrate into the neighboring blood vessels and coming into the different organs. So we got this the ketchup mice from Gunjer's group in Germany, which expressed TD tomato under the Ly6G promoter, and really carefully measured their migration pattern relatively to the blood vessels. And we, among many different days of after transfections, and we couldn't see any neutrophil actively reverse migrate to the neighboring vessel. So at least in this influenza infection case, the reverse migration may be not the major way to resolve this neutrophil. But of course, the central dogma is neutrophils come, they undergo apoptosis and clear by tissue-resident macrophage. The many of these apoptotic neutrophil clearance assay use lose-up signal type of assays. For example, you label neutrophil apoptotic cells, and whenever it clear by phagocyte, you lose signal. So this type of lose-up signal type of assay is actually quite difficult in vivo imaging, because a lot of time you want to have pulse positive or pulse negative type of assay. So we want to build an assay that actually produce gain of signal type of assay to visualize apoptotic neutrophil clearance. For that, we used a phenomenon called horizontal gene transfers. There are several investigators several years ago found that some of the gene in apoptotic cells can transiently activate in the phagocyte after they being engulfed. So if we thought that if we co-incubate apoptotic neutrophil that carried Cree genes together with macrophage or phagocyte that carry the rosa TD tomato, some of these Cree genes will be transiently activated in a phagocyte after they be engulfed, and it will induce recombination to produce signal. So amazingly, it actually worked quite nicely whenever we co-incubate neutrophils and macrophage. We very often see very active red color developments in this assay. And this is only happening whenever we co-incubate rosa macrophage with rhizome Cree neutrophil. And even in vivo setup, if we inject apoptotic neutrophil that carry rhizome Cree genes into the rosa mice, after several days, there are many red cells discovers, and 100% of these red cells were macrophage marker positive. And you can actually track the same spot in multiple days. So within 48 hours after injection, there's a lot of neutrophil remain, and none of red color cells discovered yet. By 48 hours, many neutrophils disappear, and we have some green signals start to show up. And by 96 hours, almost all neutrophils gone, and we have more obvious red color development. So that really suggests that, at least in our setup, that the many neutrophils come and they clear by tissue-resident phagocyte. So encouraged by this data, we decided to look at the neutrophils and potential phagocyte simultaneously in vivo. So for that, we crossed Ly6G mice with the CSF1 GFP mice. So in here, neutrophil express TD tomato and GFP, so they are orange colors. And all the potential phagocyte, macrophage and monocytes are green cells here. So with that, before infections, of course, there are going to be many tissue-resident macrophage, which only express CSF1R, and very small neutrophil obviously will learn from EVAN. There must be some residual neutrophil stay in the trachea. But if we infect these mice, of course, there's massive infiltration of neutrophils coming into tissue and massive monocyte coming in. But we always have this mid population, Ly6G TD tomato mid, CSF1R GFP positive. But the majority of this population doesn't actually express cell surface Ly6G, but instead they are inflammatory monocyte positive cell population. So if you look at the image here, there are many neutrophil and many macrophage and monocyte, but there's some cells that carry these small red dots. And they're presenting this mid population here. And these are cells that, as you can see, just engulf this red fluorescence expressing neutrophils, and then they processing these signals inside. So this data suggests that at least inflammatory monocyte is actually a very important phagocyte for the apoptotic neutrophils in this setup. And it is actually the case if we actually infect mice, the CCR2 knockout mice, which doesn't produce circulating monocyte, the neutrophil clearance and apoptotic neutrophil clearance was very delayed in this mouse. So then, the conventional, again, the consensus here is that the many apoptotic cells die and they kind of stay and release E-me and Phi-me signals, and this phagocyte actively approaching those apoptotic cells and eat them. But it is not the case in our system. Instead, it was the always neutrophil that actively searching for their phagocyte. So in this case, you can see this neutrophil, and none of this phagocyte is very active, but neutrophil is actually searching in between this very static phagocyte multiple times. And it is always the case that, as you can see, it is the neutrophil approaching to this phagocyte, and they being engulfed. So our data really suggests that in this case, this apoptotic neutrophil is really searching for their own phagocyte, and it's like almost kind of find your own phagocyte model. So after we've seen this, what's the conclusion of this clearance, right? So the inflammatory monocyte engulf apoptotic neutrophils, and they might be potentially, producing many anti-inflammatory, molecules to increase the resolution of inflammation. But none of those pro-inflammatory or anti-inflammatory cytokine change in the absence of neutrophils. So maybe it's not the main pathway, but only marker that change on this inflammatory monocyte if there's no neutrophils around. It is the DC differentiation marker. So if there's no neutrophil in the tissue, the inflammatory monocyte cannot express DC differentiation marker. Why this is the important? Because when T cells get activated in draining lymph node, once they arrive at the tissue, they have to encounter with tissue-resident antigen-presenting cells to boost their effective function. And it is the case that in normal infection, many T cells come into the tissue and they make very stable kind of synapse with the CCR2 positive antigen-presenting cells in the tissue. But if you deplete neutrophils and then infect mice, none of these T cells can make very stable interaction with CCR2-driven antigen-presenting cell at the tissue. So that really suggests that apoptotic neutrophils help inflammatory monocyte to become a better antigen-presenting cell for T cell activation. How? So it's been known that GM-CSF and IL-4 and MCSF is a key cytokine for monocyte differentiation into DC, but none of these things change in the absence of neutrophil. But again, only one growth factor or cytokine, in this case, which was EGF, was completely disappear whenever we deplete neutrophil from the tissue. And surprisingly, neutrophil express a lot of EGF, but none of known inflammatory stimulators can trigger neutrophil to release EGF. But it is only the apoptosis can dramatically trigger neutrophil release EGF, and it was completely depending on caspase activity. So the conclusion was, during apoptotic cells, apoptotic neutrophil clearance, they release important EGF, and that can help inflammatory monocyte become a better antigen-presenting cell that localize at the tissue for proper T cell activation. So the resolution or clearance of apoptotic cells is not just simply clearing cell number, but it actually has very important functions for host immune response. With that, not only early neutrophil response, but also the resolution of neutrophil is actually quite important for host immune response. And many of people in the lab contribute to this study. The main Ki-hong has been a major driver for this study. And I'll be happy to answer some of your questions.
Thank you very much, Dr. Kim, for a really interesting presentation. So I'll start by asking, do you think that what you're dealing with in this model is kind of like an idealized, perfectly calibrated response to infection, right? Because I'm just wondering whether you can comment on this kind of conventional wisdom there, where neutrophils often play a detrimental or destructive role in airway infections, right? Is this something you would see only when the response is really dysregulated? Or is it something you don't believe is happening at all?
I think that's a very important point to emphasize. I think that the neutrophil response has always kind of, it's a two sides of the coin, right? So you do need neutrophil for proper immune response, but delay in neutrophil clearance or prolonged neutrophil present in the tissue is always detrimental for the host. So I think this resolution is actually quite important because, I mean, that's why, the proper resolutions and controlling this kind of yin and yang of neutrophil response is critical. And I agree, I think it's mostly the intensity of inflammation infection will be dictating that sort of threshold. So that's why, the more understanding of this proper neutrophil resolution is actually going to be very important to understand that threshold.
Okay, thank you. There's a question about neutrophil extracellular traps. Do you see them in the system, or would you expect to see them only beyond this threshold that you just referred to?
I'm sorry, repeat that question again.
Question about NETs, neutrophil extracellular traps, where you managed to, have you observed them in your system?
So, of course, yeah, the NET was our first sort of speculation when I saw these particles, and we really try hard to stain, like, the chromosomal DNA staining, and it was completely negative. So again, we cannot exclude the NET in certain stage of this neutrophil migration, but at least for trail formation, NET was not the major contributor.
Okay. Next question is, it says, excellent presentation. Can you comment on the nature of the trails neutrophils are leaving behind? There's
so many nature in this trail. So one thing I comment is we thought originally it could be an exosome. Was not the case. Was not active process. It was actually integrin-dependent passive detachment, and the many of trails and particles completely closed structures. It contains a lot of cytoskeleton molecules and some other molecules other than chemokine. I mean, I can explain so many details, but that's kind of the very brief summary of the nature of the trails.
There's also a question about your horizontal gene transfer setup. Would it be possible to do that in vivo during the infection?
Oh, absolutely, yeah. That's so. It was actually kind of striking. I mean, we thought that it would be great for the assay for following the fate of specific cell type. But at the same time, it's actually kind of scary because now different type of genes actually transfer into different type of cells, and they're going to confuse a lot of genomic process. So I think for that it's going to be also very important to follow up. But yeah, I agree. I think it's going to be great tool. One thing I need to comment is it doesn't represent 100%, so it's not quantitative assay. just, you know,
Mhm.
but you can say sort of yes or no type of answer. Yeah.
Okay. Moving on to the next question. Excellent talk, Dr. Kim. Could you see viral load has any detrimental effect in apoptotic neutrophil clearance?
I mean, so again, like our model is 100% survival model, so it's just not, you know, the very severe disease, but you know, a lot of severe viral infections, and even in human case, the many patient is not dying because of the virus; they actually dying over activation or inflammatory response. So I think it's a kind of sort of working together. You know, the viral titer and also the host immune response kind of work together and make things severe. So that's why I think it's really complicating to answer that kind of question. So yeah.
There's also a question about the contribution of reactive oxygen species such as hydrogen peroxide and the enzymes that generate them, whether they participate in the response.
Yeah, of course, whenever you see neutrophil, you have to consider that. But I thought, like, you know, the ROS or reactive oxygen responses must be more important factor in, again, severe inflammation like sepsis or some viral bacterial infection. Again, in mouse models that we use is kind of sort of mild influenza infection models and recovery models. So we couldn't really address that in our model.
Okay, the next question attempts to link the first and second talks, and it's about CXCL12. So we heard before that neutrophils can be attracted to CXCL12. Do you think that after being attracted to inflamed tissue, they acquire this property of releasing CXCL12 on their trails?
Yeah, I mean, so we don't, you know, I think that's a good question. According to Ivan's data, I mean, obviously they express, you know, CXCL12 at an early stage, and one thing I can say here is the CXCF was not in their primary secretory granule, not in the secondary, or not in the tertiary granule. So somewhere else. We don't know. And I mean, because like I said, you know, neutrophil express many different chemokines, and why only CXCL12 does this, we don't know. I mean, this is a very important question, but we don't know the answer yet.
So can I comment on CXCL12? Transcriptionally, we found also CXCL12 RNA in neutrophils. So yeah, that makes sense to me. I mean, we found it as a signal, but they can also produce it, I think, also. Yeah.
Yeah, or maybe, you know, this can be mRNA, you know, micro exosome or type. Yeah. Yeah. Thanks.
Good. And then last question is about the trails and whether they could be related to emperipolesis between megakaryocytes and neutrophils. So is there a role for megakaryocytes in this?
That'll be an interesting question. Maybe I should talk about that with some expert in the field. Megakaryocyte. So again, you know, we don't think this trail is actually active process. I think that, you know, a lot of sort of megakaryocyte and their exosomes and platelet production actually is a very active process. But one thing I can comment on that is the many cells actually make trails. It's not only the neutrophil, even T cells, we see a lot of trails during migrations, and even cancer cells leave trails. That doesn't mean that all these trails have the same function in different cell types. So, you know, right? Different cell type might have very different function for their trails. So